Okay, good morning, everyone. So let's continue our discussion today about how our cells, eukaryotic cells, have different opportunities for controlling how genes are expressed. And importantly, we'll keep thinking about this idea that all the different types of cells in our body, right, they all have a copy of our master genome in them, with the 25 or so thousand genes that make up a human being. And how is it that the different types of cells in your body have control over what subset of those genes they actually use in order to be the type of cell that we need them to be? So last time we began this discussion, and we're going to go back to this particular slide a few times over the course of the next couple of lectures, because this shows us sort of the 5 main levels of opportunity for a cell to be able to control whether a gene is expressed. And last time we spent time looking at how we can, on a very fundamental level, simply control whether or not genetic sequences in the DNA that represent our chromosomes are simply available in order to be read, transcribed, and expressed to make protein products from them. We have a couple examples here of different ways that at the chromatin level, a cell can control whether a gene is accessible. There are a couple of cases where you can actually imagine the actual sequence of the DNA being kind of altered. For example, you can, in rare cases, certain types of cells can make copies of genes so that you have multiple copies of the genes, therefore increasing the amount of product that comes from those genes. So gene amplification and deletion, we don't really need to talk about that too much because that's a very specialized rare case. Same thing with DNA arrangements, having different fragments of genetic code on a chromosome that can be rearranged to make functional genes. Those are used in things like certain immune cells when we're making things like antibodies. When our B cells make antibodies, they rearrange genetic sequences in order to come up with new protein shapes. Right? That's how you develop new antibodies that you didn't have before. But those are kind of rare cases. When we think about the majority of genes in our genome, they're controlled by processes such as DNA methylation, the physical condensing or relaxing of the chromatin, modifications to histone proteins, such as acetylation or methylation of histone proteins that then relax their grip on the DNA so that the DNA can unwind a bit from the nucleosomes. And there are some examples of RNA products that are transcribed from genes that ultimately serve to control the regulation of other genes. So in order to finish up, last time we ran out of time, and I didn't get to this discussion of DNA methylation and non-coding RNAs. So that's what we'll start off today, just to kind of finish up where we left off. Keep in mind that these things are all sort of acting on the chromatin itself, ultimately to allow a gene to be available for expression. So DNA methylation, what is it? It's just what the name suggests, right? It's the addition of methyl groups, CH3, to the bases of the DNA itself. Okay. So in our bodies, in eukaryotic cells, there are certain pairs of nucleotides, C's followed by G's, in other words, a CpG dinucleotide. Right? So this would be on the same strand of DNA. It's two neighboring, a cytosine and a guanine that's linked by the phosphodiester bond. And that's why we denote it as a CpG, so that we know we're talking about two bases on the same strand. Those cytosines can be recognized by a family of enzymes known as DNA methyltransferases, or DNMTs. The shape of the DNMT protein, so this gray blob, is this DNMT protein. It is the right shape to fit around the double helix of the DNA, and specifically, it's the right shape so that when you have a CG next to each other, the DNMT1 enzyme can transfer a methyl group onto the cytosine. So here's the cytosine originally. Once the DNMT enzyme or the protein is wrapped around the DNA, it puts a methyl group onto it. And what you end up with is now 5-methylcytosine, right? So it's a cytosine that has a methyl group at the 5-carbon position. What does this do? Well, it alters the shape ever so slightly, right, of that nucleotide. And as a result, the DNA strand in that particular spot is going to have its shape ever so slightly altered. What this results in is things like, for example, the nucleosome that has this stretch of DNA wrapped around it. This slight alteration in the shape of the DNA makes the histone proteins of the nucleosome hold onto the DNA tighter. It's a better fit of the DNA wrapped around it. Right? So you can imagine then, if you have a whole bunch of CPG sites clustered together in a particular stretch of DNA and they all get methylated, that particular spot of DNA might be wrapped around a nucleosome much tighter than other sites. So this provides a mechanism by which the cell can selectively condense or make heterochromatin in certain regions. So where would these regions be? Well, they would be regions that contain certain genes that are being controlled this way. If you look at, let's start off here looking at this set of sequence on the right. So this is a typical non-promoter DNA region, right? This would be like if you closed your eyes and just put your finger on a chromosome somewhere, chances are that wouldn't be the promoter of a gene. That might be somewhere way in between two different genes. It might be right in the middle of a gene. Right? And so in this type of a sequence here, what they've done is in yellow, they've highlighted where you see CPGs, right? C's next to G's in the sequence. So each of these is a C and a G. So statistically, what might be surprising is that those CPGs appear far less frequently than you would anticipate by chance. Right? In other words, what are the chances of having a C next to a G? Purely by odds, it'd be a 1 in 4 chance times a 1 in 4 chance, a 1 in 16 chance, right? So every 16 pairs of bases, you maybe expect to see a C next to a G. But what you find is that's not the case. Right? Over evolutionary time, we've lost many of these CG positions. But that's if you're looking in a non-promoter region. On the left, this is what you see in a typical promoter of a gene. And you can see there are far higher numbers of these CG dinucleotides in them. Right? In fact, here you see an ATG, so this would be the translation start site eventually of this gene after this has been transcribed, right? So this is the promoter region of it. And what you find is that in many cases, you have what we call CPG islands, several CGs together. Now you can imagine, right, that if the cell wants to shut down the promoter region of this gene, it can methylate all these C's that are part of the CPGs, right? And then this region of the chromatin would become very condensed, tightly wrapped around nucleosomes, and you would no longer have the promoter be accessible for things like RNA Pol2 to come in and transcribe things. So let's think about this. What we're talking about is this situation down here, where these little red dots represent potential CPG sites in the DNA sequence. The DNA is the black strands, and it's wrapped around all these nucleosomes. Right? So the methylated cytosines, so these 5-methylcytosines here, cause all these nucleosomes to really tighten their grip on the DNA, and this whole promoter region becomes heterochromatin. Right? It's too tightly packed. The genetic sequence of the DNA is no longer available for things like transcription factors to come in to recruit RNA Pol2. However, we did discuss that the histone proteins that make up these nucleosomes around which the DNA is surrounding, right, they can be modified to both tighten and loosen the grip on chromatin. So here's a situation where you can see the CPGs are now not methylated. So they're white dots instead of red dots. And in addition to that, these green stars represent acetylation of the histone proteins that are also now releasing their grip. So these are like the two extremes, right? This top one would be euchromatin, chromatin that's been unwound so that now the DNA sequence can be read, so proteins can come in and bind to it, ultimately starting transcription at this promoter, whereas the bottom would be a promoter that's really shut down, right? And therefore, the cell that has decided, so to speak, to methylate this promoter, right, no longer has access to this gene. And it might be that that's on purpose, because maybe this gene is a gene that's not relevant for this cell in order to carry out its normal functions. What about this idea of small RNA transcripts playing a role in gene regulation? I think a classical example of this is the phenomenon of X inactivation. So remember, female mammals, whether you're talking about humans or this cat, right? Females inherit two X chromosomes, one from mom and one from dad. Evolutionarily, you only need one X chromosome, because the genes that are on that X chromosome, we've evolved to only expect to have one copy of them. I mean, after all, males, right, only have one X chromosome. Right? So in females, one of the two X chromosomes needs to be shut down. And when I say shut down, what are we talking about? Becomes so condensed that none of the genes on that second X chromosome are accessible. These shutting down of one X chromosome or the other is pretty much done at random by stem cells as they are dividing and starting to become the different cells that make up the different tissues of the body. So very early on in the embryo, these different stem cells that make up the embryo start making this decision at some point in a female embryo, which X chromosome to shut down. So we can see this very classically in cases of the calico fur pattern in cats, right? Calico cats are ones that have these patches of, let's say, dark brown or black and orange, right? So it turns out that the gene that has an influence on fur color of the cat is located on the X chromosome. So let's say this embryo of a cat got the orange allele from, I don't know, mom, right? And the black allele from dad. Right? So depending upon which one of these is allowed to function, eventually the fur will be black or orange. So as this early embryo is made up of many, many stem cells, in these stem cells, they randomly start turning off or on. One keeps the orange one and turns off the black one. One keeps the black one and turns off the black one and keeps the orange one. And so once these stem cells finally create all the different parts of the cat, you actually see modeled fur color. So in other words, these patches of the cat's skin that are orange are ones where originally the stem cells that made these kept the orange allele, right, but shut down the black one. And the black patches of fur are the opposite. So how does this happen? How does one X chromosome get shut off and the other left on? It has to do with expression of a small RNA transcript that comes off of a gene known as exist. And so what happens is on one of these chromosomes at random, the X chromosome, it begins to express this exist RNA, which are these little green squiggles here. And this RNA, it doesn't get translated into a protein. It just functions as a small RNA, and it begins to coat the entire chromosome. And when it coats the chromosome, that coating of RNA attracts a particular protein to cover this chromosome, and that protein results in the chromatin of the chromosome becoming super, super condensed, creating what we call a bar body, right? The bar body is one of the two X chromosomes that was deactivated. This happens in a cat. This happens in a person, right? And here you see that here is an example of one of the X chromosomes covered with this exist RNA. Right. So I think it's a fascinating mechanism because, again, it involves expression of a particular gene that doesn't end up making a full-blown protein, but instead function just as an RNA molecule that does something very important. All right. So that brings us to the end of what I had hoped to get to last time. So we finished that up. Ultimately, what we've been talking about are different ways of modifying the chromatin just to make a gene available or unavailable, right? That's kind of the most fundamental level of how a cell can control whether that gene gets used. So we're talking about here, this ball of yarn. Here we have DNA that's now available, right? And that brings us to the next level of control that we're talking about today, which is, okay, now that you have regions of a chromosome available, right, as euchromatin, unwound, so now the DNA sequence is visible or available. Now, right, how can you control whether a gene in that region gets expressed? This has to do with all the proteins that are now involved in whether or not RNA Pol II complex, this big protein complex, ultimately is able to sit at the promoter and start transcribing the gene, all right, in order to make the primary RNA transcript. Right. So transcription is controlled by a whole bunch of different proteins that function as transcription factors, right? We said before, transcription factors are proteins that are the right shape to bind to DNA, and by doing so, they kind of set the stage for something else to happen at that particular spot. Ultimately, a pile of transcription factors at the promoter will be the right shape to attract the RNA Pol II complex, right? But there are many events that lead up to that, okay? And that's what we're going to take a look at. The promoter of a gene alone is not going to give you fine enough control to explain how it could be that out of 25,000 genes at a cell's disposal to use, the cell will only use a particular subset of them, right? Because all genes have a promoter, right? So when we look at this here, or we said the promoter is that region where you have different transcription factors that are going to bind, forming a little pile right at the sort of core part of the promoter, right? Ultimately, that pile will be the right shape for RNA Pol II, the big purple thing in the back, right, to be attracted and bind here and fit. And RNA Pol II will start transcribing right at the transcription start site, right? So this big pile that makes the right shape for the RNA Pol II to come along are your basal transcription factors. In other words, these are kind of generic transcription factors that recognize sort of generic conserved sequences that define a promoter. Right? However, all cells would be making these purple basal transcription factors because they're needed at every promoter. So that alone is not going to give you fine control, right, over whether this gene gets expressed, but this one doesn't. So that means in addition to these core promoter sequences, right, and these core promoter transcription factors, there must be additional DNA sequences surrounding the gene that all contribute to what's happening at the promoter. Right? And so these, not surprisingly, would be other stretches of DNA sequences that also attract different transcription factor proteins, but in physical, either close by the promoter of the gene that they want to act on, or sometimes very far away. We call these regulatory sequences that give the cell's ability to sort of adjust what's actually going on at the promoter in a very finely tuned way, depending upon what the cell needs. Let's look at this analogy to kind of illustrate it. Let's think about a very busy airport, right, where you have dozens of airplanes at any given time, landing, taxiing, taking off, etc. Right? Let's pretend or imagine that each individual airplane is a specific gene, right? So its activity, right, needs to be tightly controlled, because otherwise, how could this airport avoid disaster? Each one of these airplanes has its own core promoter. That is the cockpit of the plane itself. And so you might say, yeah, so the most important elements here are the pilot and co-pilot, right? Because they ultimately have the most sort of intimate control over what's going on with this airplane, right? So You cannot just expect, though, that because every single one of these airplanes in the picture has a pilot and co-pilot, that that's all you need, right? Because how would these pilots and co-pilots know what they're supposed to be doing, right? They would just sort of on their own decide whether to take off or land or taxi to that gate or go on this runway, right? You cannot have that. It needs to be a very coordinated effort, right? That's why you need the control tower. Right? So in the control tower are different important people who are there as, let's say, radar technicians, the radio people, people who are talking to the maintenance crew, the ground crew, people are talking to the terminal gates to find out what's going on, right? People who are just like with binoculars looking around. And these people all have to be recruited and they all have to work together in order to do what? In order to properly coordinate the activity of all the airplanes at one given moment, right? And they do that by talking to the pilot and the co-pilot of each of the airplanes, right? So if we think about every gene, right, is going to have regulatory elements overseeing what's ultimately taking place at the promoter. Just like at this busy airport, you're going to have a whole bunch of specialized technicians in the control tower overseeing everything, ultimately telling the cockpit of each airplane what it should be doing. Right? And when I say specialized people in the control tower, what am I talking about? I'm saying each one of these represents a different type of transcription factor that has to be brought together so that together they create the right set of signals in order to communicate with the promoter of a gene. So transcription factors. Let's take a closer look at how transcription factors work. I find this fascinating. So actually much of my research has to do with how transcription factors function in certain types of situations of diseases like cancer. So remember, transcription factors, the most kind of simple definition of it is it's a protein, that it's the right shape, that's designed to sit on DNA and do something that ultimately is going to influence transcription of a gene. So we call transcription factors TFs, right? And they're going to bind to the DNA by recognizing certain short specific sequences of A's, T's, C's, and G's in the DNA that make the DNA the right shape in that spot so that the right shape of this particular transcription factor can fit there very nicely and very securely. Here's an example. There's a transcription factor known as BAF, right? And the function of BAF is to loosen histone interactions. So here you see a model of the DNA wrapped around a nucleosome. So in this model, the DNA would be coming from behind you, going towards the screen, right? And then it would be winding around. So let's say it comes from behind you and it comes here and it's winding around this nucleosome, 146 base pairs of it go around the nucleosome and somewhere it then continues on, right, into your screen to the next nucleosome, right? So this is like a cross section of it. So here are the histone proteins at the core of the nucleosome. So here's the BAF transcription factor. Here's the ribbon diagram of it showing the peptide backbone. Here is the space filling model, right, showing the three-dimensional shape of it. And so you can see it is just the right shape. It has these alpha helical structures in its protein secondary structure that perfectly fit in the grooves of the DNA, right? And so that contact point between those parts of the protein and the DNA will be influenced by what the nucleotide sequence is of that DNA. You don't think about this a lot. You think that no matter if you have A's, T's, C's, or G's, the DNA is still shaped like DNA, right? Yeah, you still get the correct angles of the double helix, things like that. But in between each of the twists, where you see the rungs of the ladder exposed, think about it. Whether you have an AT there or a CG there or a GC or a TA, the shape is going to be ever so slightly different in there, right? Almost like the little texture of the three-dimensional shape in between, right? So therefore, certain sequences are exactly the right shape to be a good match for these proteins. One way you can determine what are these DNA sequences, right, that BAF actually binds to is you can do studies like this. Researchers have looked all through the genome in cells to identify where they can see BAF protein stuck to chromosomes. Right? So let's just say, for argument's sake, here's 3 examples of chromosomes, and here's maybe like 1, 2, 3, 4, 5, 6 different spots where the BAF protein can be detected binding to DNA. What you do then is you just go ahead and you sequence the DNA in those spots. So let's say you don't do this 6 times, but let's say all through the genome, you find 1,000 locations where BAF binds. You sequence all of those regions and you build a consensus. What sequences seem to be, right, the most common as the ones where BAF binds? And by comparing these sequences, let's say 1,000 spots where BAF binds, you can build what we call a consensus DNA binding motif. What you see here is, let's say you have 12 bases that are the DNA bases directly under where BAF is binding to the DNA, right? And after looking at 1,000 different BAF binding sites and comparing, is there a T there, an A there, a G, or a C? This is like voting, right? 1,000 different votes, and these are the winners. So in other words, of these 12 DNA bases at the binding site for BAF, at position, what is this, 4 over here, nearly all of them you find an A there, right? Or at position 7, nearly all of them you find a T there. Whereas at other positions, such as position 2, maybe a third of the time you find, a third of the 1,000 sequences that we looked at where BAF binds, there's a T at position 2, and another third of them have a C there, and maybe roughly a third of them have an A there, right? Other sites like nucleotide position 11, it seems like it doesn't really matter what you have there. Right? But this is neat, because what this shows you is that this would be the ideal sequence in the DNA to be exactly the right shape, so that the BAF protein, because of its shape, can fit in there, right? Like a key fitting a lock. So what does this have to do with how different cells might control different genes, right? So in a case like this, you might say, yeah, if a cell makes a lot of BAF protein, that means all of those different genes that have this sequence in the DNA that BAF likes to bind to, maybe those genes as a whole set would be upregulated or turned on. Hopefully this gets you thinking, right? So depending upon what important transcription factor proteins you have in a cell, that will influence what sets of genes are being turned on in a wave, right? Or shut down in a wave. Here we have an example, I think, that illustrates this even more. We have 4 closely related transcription factor proteins here: OCT2, engrailed, MSX1, and PRH. They're all part of the same family of transcription factors because in ancient evolutionary history, they probably all started as the same gene that then got duplicated and were put on different chromosomes in the genome. And as a result, over time, they all mutated. And now, as a result, the protein product that comes from them is slightly different. Right? Here is the amino acid sequence of those proteins, specifically just looking in the region where they bind to DNA. And these are the amino acids, right? So hopefully you remember these are amino acid abbreviations. In the secondary structure, once this series, this polypeptide chain folds up, it forms 3 helical regions, helix 1, 2, and 3. And this is what you get. These are the 3 helixes, 1, 2, and 3. Of each of these 4 example transcription factors shown in their corresponding color. Right? And they've overlapped them to show you how closely related, how similar the final protein is. The helix 1 and 2 is in charge of getting the protein sort of nestled up against the DNA, which is the red thing here. Right? Helix 3 is the one that lays across the major groove of the DNA. And what you can see is that very clearly, all 4 of these proteins, they overlap so well in position. Clearly, they're from the same family of proteins, but because of slight differences in their genetic sequence that codes for these proteins, right? You get slightly different amino acids at different positions. So in these helix 1, 2, and 3 amino acid sequences, where you have conservation or the same amino acid shared among the 4 transcription factors here, right? It's colored here. But you have variations. For example, this position here, each of the proteins has a different amino acid there. What does that mean? That means the actual helix 3 shape of the proteins is highly similar, right? Because they share the same amino acids. You can see here, the shape of the helix 3 is conserved. Why? Because it has to be conserved if it's going to fit so well, right, into that major groove. You're not going to have a lot of variations, crazy changes in the amino acid sequence, but variations in the other amino acid positions result in the overall shape of the protein being a little different, so that the position of the helix 3 might be a little this way or a little that way. So that's when you have to look at the nucleotide sequence of the DNA. So here's a space-filling model of the DNA, right? And here what they've done is in orange highlighted the particular atoms that end up interacting with the helix 3 of this family of transcription factors. So some of these are the phosphate backbone, right? So this black dot is the phosphate group. And so these orange things are maybe like the oxygens sticking off of them. But some of these, like the ones right here, are clearly part of the particular nucleotide. So depending upon if you have an A here or a T or a C or a G, right, these orange atom locations in the middle would be slightly positioned differently. As a result, these 4 related transcription factors have variations in the actual preferred DNA binding motif. So that means genes in your genome that happen to have this sequence around them, they may be acted upon by OCT2, whereas this one, if you have genes that have this sequence around them, those genes get acted upon by the engrailed. MSX1 and PR, you can see the PRH here, right? They have very similar binding motifs. The only difference really being that the DNA binding motif around genes that respond to the PRH transcription factor also have this T upstream, right? But they both have this AATTA. Right? Sure enough, if you look at the helix 3 sequence up here, right, you'll see that the MSX1 and the PRH, their helix 3 is so similar. The only difference being that one of them has a serine here, right, instead of a leucine. Right? And as a result, the position of that helix 3 is going to be ever so slightly different, that maybe in the PRH one, you really need to have a T in here as well in the DNA in order to get it to fit correctly. So different cells express or have different sets of transcription factors in them that then determine what subset of genes that have these particular motifs around them will be turned on or kept shut off, depending upon what the cell needs. Okay. So these gene regulatory elements then would be sites that are around the genes where transcription factors, like the ones we just looked at on the previous slide, start piling up, right? And if they pile up in a certain way, then they are like the control tower at the airport. They can send signals to the promoter of the gene telling the cockpit crew of the airplane, hey, you should taxi around now, you're free to take off, or you should stay grounded, right? In other words, whether RNA-Pol2 ultimately transcribes the gene to express it, or RNA-Pol2 doesn't go there and the gene is kept turned off. Okay. So these regulatory sites then, they have different types of functions. Some of them can be stimulatory, some of them can be inhibitory, right? Some of them can serve to actually block interactions between other regulatory sites and the promoter. So we're going to take a look at these here. Some of these sites can be close by, some of them can be far away. Right, so the 4 main sites we're talking about are proximal regulatory elements, enhancer elements that can be far away, silencers that can be very far away that repress activity at the promoter, and insulators that can actually block the signals from other regulatory sites. Let's take a look at how these things might be arranged. So here you have a gene, right? So this gene, the transcribed part of the gene is in blue here. So it's a combination of exons and introns. right? So here would be what we call the core promoter region. So this is where transcription starts. So you can imagine this is where RNA Pol II eventually needs to come and be recruited here in order to start transcription to make the RNA primary transcript. Right? So that's the core promoter. So it has some sort of generic things like a TATA box, where TATA binding factor would bind, that protein would bind here as part of helping to pile up a shape that RNA Pol II would be attracted to. Right upstream of it, right, you have these proximal or nearby elements. You can see this CAAT box is 80 nucleotides upstream from the transcription start site, right? Transcription start site is +1. You have this CG box that's about 100 bases upstream of the transcription start site. So 80 or 100 bases away, that's still considered pretty close, right? So that would be like if this is our airplane and we say the core promoter is like the cockpit, these two boxes would be like the ground crew, you know, standing in front with those, I don't know, those flashlight pylon things, right, waving them around, right? Still, it's pretty immediate, pretty intimate control over the promoter. So those, all of this stuff, like the proximal elements and the core promoter, let's just say that's all part of this green region here, right? Now, if you follow the DNA, not just 100, 200, 500, but maybe like 5,000 or even 50,000 DNA bases upstream, very distal, right? Not proximal, but distal. Now you're talking about stuff that's happening up in the control tower. So an enhancer region would have DNA sequences that attract different transcription factors to bind there and pile up. And ultimately, that signal will stimulate the recruitment of RNA Pol II at the promoter so that this transcription level of RNA Pol II making RNA goes up, right? And it's enhanced. Right? You might have regions like the silencer, right? A different region than the enhancer that also recruits transcription factors that recognize their DNA like consensus sequence, but their signal inhibits RNA Pol II from being recruited or from RNA Pol II being allowed to go ahead and start transcription. So they inhibit or repress transcription. Then you have regions like this insulator one that again recruits its transcription factors to its DNA sequence, but you can see what it's doing here is it actually gets in the way of these other regulatory sites from controlling certain promoters. In this particular case, you have two genes here, one that has a promoter over here that gets transcribed off to the left, another gene, gene number 2, its promoters over here, right? It's normally would be transcribed to the right. This enhancer has bound all kinds of transcription factors that then signal this gene to go ahead and transcribe. But this insulator in between this enhancer and this promoter of gene 2 gets in the way of those signals, right? So this is very fine control. Right? This is actually determining which gene is allowed to transcribe and which one does not. So when we talked about the analogy of the radar tower or the control tower at the airport talking to the individual cockpits of the airplanes, you know, that's like radio signals, right? So here you can imagine that these black arrows, whether stimulatory or inhibitory, are like the radio signals. But of course, you know, in your cell, you know, there's no radio signals, right? So how is it that the enhancer, silencer, or insulator actually does their job? Well, we're talking about piles of proteins that have been coordinated in these different staging areas so that when the right combination of proteins come together, right, they can then do something. So the way they do something is always based upon what? It's always based upon shape, right? The right shape piles of proteins will then be able to communicate with the right shape piles of proteins forming at the promoter. So that means that you actually have physical interactions of these different regions of the DNA. And that's precisely what happens. We tend to think of DNA in a very linear fashion, right? But that's kind of naive because here we have, again, a part of our chromosome laid out from left to right, but we already know that DNA is not a rigid line, right? It is a long molecule that's free to wrap around. Last time I showed you pictures of a bowl of Simon, right? I got you thinking about how the loose noodles are like our chromosomes, right? So as a result, just like one single noodle in your bowl of noodles of your Simon, yeah, right? There are different parts of that noodle that might actually contact itself as it loops around, right? And so that's what happens. Here we have, let's say, a gene with its core promoter, and somewhere very far upstream of it, maybe 5,000 or 50,000 DNA bases away, is the enhancer, right? So in the linear sequence, these two regions you might not think have anything to do with each other, right? But in true three-dimensional space, they may interact. So for example, this enhancer has these yellow sequences that attract these two different activating transcription factors, right? These are slightly different shapes. Each one binds to its own consensus DNA sequence there and begins piling up other proteins that aren't necessarily shown here, right? You have different other proteins that are also attracted here. Ultimately, right, the shape of this pile that's forming at this enhancer region, right, which we call an enhancosome, which just means all the proteins that are going to be involved, they're piling up, and that functional pile of proteins is the enhancosome, is going to recruit other proteins such as this SWI/SNF or this hat that does functions. Like the SWI/SNF, for example, ends up altering the spacing of nucleosomes from each other on the chromatin, or the histone acetyltransferase hat starts putting acetyl groups on histone proteins to loosen the activity. Right? So these are recruited here by the enhancosome, and this encourages the DNA to loop around so that that big pile of things that were getting prepared in this staging area, right? I call it a staging area because it's kind of like when you go to a play or a movie, right? Currently the scene that's being done, you have the actors on the front of the stage, and then you have the curtains, and in the back, you have all the people frantically putting together the next sets or changing clothes or whatever, right? It's like a staging area. And then when the time is right, those people come out. So when the time is right, these proteins that are piling up at the enhancer end up recruiting a particular protein known as a mediator complex, and that brings their activity over to the promoter. And that then encourages, right, these basal transcription factors that have just been floating around now to be attracted here. They pile up, ultimately recruiting RNA polymerase that begins transcription. Okay, so let's visualize how this might look, not just as a cartoon, but something more realistic. Right, so here's a typical eukaryotic cell. If we zoom into the nucleus, I showed you a picture like this before when I was using that analogy of a bowl of Simon, right? And we saw that there are different regions in the nucleus. The nucleus is filled with noodles, right? All the different chromosomes that are unraveled. Certain parts of it, they're more dense. So these darker blue, or these, I guess these brighter blue spots might be denser chromatin. The more diffuse, sort of speckled blue regions might be the loose noodles, right? Here, what they've done is they've actually highlighted one individual chromosome, right? So this is one individual chromosome all unraveled, and it's sort of in this little spot in its own sort of territory within the nucleus. This is awesome, right? Because what they've done is they actually generated the true three-dimensional positional map of what this one chromosome looks like in this cell. And this is it, right? So this is amazing. Obviously, this is done with a computer that reads all the positions and coordinates of different types of probes and things put in there. But this is an actual chromosome and what it looks like when the cell is busy utilizing certain genes from it. You can see the middle part, right, is condensed. So maybe it contains genes that the cell doesn't need to be expressing at this moment, but different parts of it are looping out, right? And these looped out regions of more euchromatin are regions where certain genes are needed at that moment, and the cell needs to make the products of those genes. Notice also, right, you can really see how this is all just one chromosome, so this is just one molecule of DNA, right? But different regions of it, you can see, are looping around, so you can have different regulatory elements brought to the promoters of different genes. And what's awesome about this is you can imagine that as the needs of the cell changes, right, this would be a dynamic structure that shifts with different parts or different loops of the chromosome coming out as other parts go in. Right? All right. So ultimately, that's the secret to understanding how we can have 220 different cell types that make up our body, right? All starting from the original genome that mom and dad gave you. Right? So here we have 3 different cells. Let's say these are very different cells with different types of functions, therefore different needs in terms of which genes from the master blueprint they actually need to follow. Right? All of them have this particular gene in them, right? Because that's genomic equivalence. All 3 of these cells in your body have the same gene in them because they have your same genome in them, but they might utilize this gene very differently. So in this case, let's say these are like skin cells, right? Maybe this particular gene has to do with coding for a protein that manages cell-to-cell contact, right? And in this particular case, this protein can just be transcribed by having RNA polymerase and some general transcription factors present, right? And that's enough. They sort of occupy the promoter, and you have a trickle or moderate basal level of gene expression. So you make this much of the protein product from it, this gene eventually, right? Whereas you have this cell, this nerve cell, and nerve cells really need to have tight control over cell-to-cell communication. So maybe this exact same gene in the nerve cell, right, you need to have a lot more of this protein made. How does the nerve cell do it? Because it has in it higher levels of this yellow transcription factor. This yellow transcription factor binds to some sort of a regulatory region, right, that enhances the production of this protein in this cell. And this is a yellow transcription factor that the skin cell doesn't bother having in it, right? So already you can see that's how the nerve cell is able to have some control over this. Then you have these red blood cells. As the red blood cells are forming, maybe they have very little interest in cell-to-cell communication. And in fact, maybe cell-to-cell communication would be bad for a red blood cell because then the red cell might clump together, right? So not only do you not make the yellow transcription factor protein in these cells, but the red cells make this red diamond one, right? And this red diamond one sits on a repressor regulatory site that really keeps this gene shut down. So these red cells don't even make this particular protein. Okay. So what does that mean? That means the presence of these different transcription factor proteins drives the expression of the same gene, but differently, depending upon cell type, resulting in different protein levels. Right? Consider this. What if the protein that's being made is itself a transcription factor with different spots in the genome that it will bind to and control its own set of target genes? By controlling the expression level of that transcription factor, right, you're essentially doing what was up here. That's how you get cells that only make the yellow one or the red diamond one, right? But the key here is the next wave of gene expression that results from the different expression levels of this, blue pentagon transcription factor will make these cells even more different than each other, right? Because the next set of genes that get turned on, right, will be even more different in the red cell versus the nerve cell or the skin cell. So ultimately, that's how you went from being one fertilized egg that had one diploid copy, master copy of the human genome, over the last 20 plus years of your life, right? As cells divided and made more of these decisions about what sets of genes to turn on and turn off, that's how you ended up making all the 200 or so different types of cells in your body. And that process continues, right? Because once you have these mature cells made, the sets of genes that they use are the genes that are only relevant to being a nerve cell, a skin cell, or a red blood cell. In the same way that we said it's like going back to that example of the architectural blueprints for a house, and you have the different contractors, the plumbers only caring about the information about where the pipes go, the electricians only caring about the information for the wiring, the masons only caring about where they're supposed to put a brick wall up, right? Those are the different types of cells in your body, but they all follow the same master blueprint. They only just use the information that's relevant for them. Okay, so I'm going to go ahead and stop there today. And I will continue on next time with sort of a more colorful analogy that I was hoping to get to today, but since we're already a little backed up, I'll just leave it for the start of next time. Okay.